During prophase, the sister chromatids condense. The mitotic spindle forms and moves to opposite sides of the cell. The nuclear membrane that surrounded the nucleus breaks down, and the chromatids can move about the cell. During metaphase, the spindle fibers help to pull the chromosomes into the center of the cell where they line up. The spindle fibers are attached to the centromere of each of the sister chromatids, as well as to the chromosomes themselves. During anaphase, the spindle fibers pull the sister chromatids apart and move them to opposite sides of the cell. The result is two identical copies of DNA, one on each side of the cell. Once the sister chromatids have separated, they are surrounded by a new nuclear envelope. The chromosomes uncoil and the spindle fibers fall apart. You can tell that cytokinesis is happening because the two halves of the cell start to push in near where the chromosomes were. It looks a little like pulling on a drawstring or tightening a belt. Microfilaments, the structures that make up much of the cytoskeleton, help pull the cell membrane together. Eventually, the edges meet, and two identical daughter cells are formed. Cytokinesis works a little differently in plants, because the cell wall is too stiff to be drawn in, even by the strongest microfilaments. Instead, a cell plate forms between the two nuclei and grows outward until it meets the existing cell walls.